Hello everyone, my name is Paramjeet Singh and my topic is cell organelles structure and functions biology. So cell organelles definition. Cell organelles are specialized entities present in a particular type of cell that performs a specific function. There are various cell organelles out of which some are common in most type of cells like cell membranes, nucleus and cytoplasm. However, some organelles are specific to one particular type of cell like plastid and the cell wall in plant cells. This is the structure of human cell and plant cell. List of cell organelles. There are 24 types of cell organelles, and the first one is cell membrane, also called plasma membrane. Plasma membrane is composed of lipids and proteins where the composition might fluctuate based on fluidity, external environment, and the different stages of development of the cell. Structure of cell membrane. Structurally, it consists of a phospholipid bilayer along with two types of proteins, embedded proteins and peripheral proteins, that function in providing shape and allowing the movement of particles in and outside of the cell. The most abundant lipid which is present in the cell membrane is a phospholipid that contains a polar head group attached to two hydrophobic fatty acid tails. Functions of cell membrane. The cell membrane provides mechanical support that facilitates the shape of the cell while enclosing the cell and its components from the external environment. It generates and distributes signals in and outside of the cell for the proper functioning of the cell and all the organelles. Cell wall. An additional non-living layer present outside the cell membrane in some cell that provides structure, protection and filtering mechanism to the cell is the cell wall. Structure of cell wall. In a plant cell, the cell wall is made up of cellulose, hemiculose and proteins, while in fungal cell it is composed of chitin. A cell wall is multilayered with a middle lamella, a primary cell wall and a secondary cell wall. Functions of cell wall. The critical function of cell wall is protecting and maintaining the shape of the cell. It also helps the cell withstand the turgor pressure of the cell. It initiates cell division by providing signal to the cell and allows the passage of some molecules into the cell while blocking others. Next one is chloroplast. A chloroplast is a type of plastic that is involved in photosynthesis in plants and algae. Chloroplast contains an essential pigment called chlorophyll necessary to trap sunlight for the production of glucose. Structure of chloroplast. It is double membrane structure with its own DNA which is inherited from the previous chloroplast. The outer and inner membrane is porous and allows transport of materials, while the stroma contains DNA, chloroplast, ribosomes, proteins and star granules. Functions of cytoplasm. Most of the vital cellular and enzymatic reactions like cellular respiration and translation of mRNA into proteins occur in the cytoplasm. It acts as a buffer and protects genetic materials as well as other organelles from damage due to collision or change in the pH of the cytosol. Next one is endoplasmic reticulum. Endoplasmic reticulum is present as an interconnection of tubules that are connected to the nuclear membrane in eukaryotic cells. There are two types of ER based on the presence or absence of ribosomes on them. Rough ER with ribosomes attached on the cytosolic phase of endoplasmic reticulum and thus is involved in protein synthesis. Smooth ER lack ribosomes that has a function during lipid synthesis. Structure of endoplasmic reticulum. Endoplasmic reticulum exists in three forms: cisternae, vesicles, and tubules. Cisternae are sac-like flattened unbranched structure that remain stacked one on top of another. Vesicles are spherical structure that carry proteins throughout the cell. Functions of endoplasmic reticulum. ER contains many of the enzymes required for several metabolic processes, and the surface of the ER is essential for the other operations like diffusion, osmosis, and active transport. One of the crucial functions of ER is the synthesis of lipids like cholesterol and steroids. Endosomes. Endosomes are membrane-bound compartments within a cell originating from the Golgi network. Structure of endosomes. There are different types of endosomes are based on morphology and the time it takes for the endocytosis material to reach them. The early endosomes are made up with the tubular vascular network, while the late endosomes lack tubules but contains many close-packed intraluminal vessels. Functions of endosomes. Endosomes allow the sorting and labeling of internalized materials from the cell surface and transport of materials to the Golgi or the lysozymes. Next one is Golgi apparatus. The Golgi apparatus is a cell organelle mostly present in eukaryotic cells, which is responsible for the packaging of macromolecules into vessels so that they can be sent out to their site of actions. Structure of Golgi apparatus. The structure of the Golgi complex is pleomorphic, however, it typically exists in three forms, i.e., cisternae, vessels, and tubules. The cisternae, which is the smallest unit of Golgi complex, has a flattened, shack-like structure that is arranged in bundles in a parallel fashion. Functions of Golgi apparatus. Golgi complex has an essential purpose of directing proteins and lipids to their destination and thus act as the traffic police of the cell. Golgi complex is involved in the synthesis of other cells organelles like a cell membrane, lysosomes, among others. Next one is lysozyme. Lysozyme are membrane-bound organelles that occurs in the cytoplasm of animal cell. These organelles contains an array of hydrolytic enzymes required for the degradation of various macromolecules. There are two types of lysozymes. Primarily lysozymes containing hydrolytic enzymes like lipases, amylases, proteases, and nucleases. Secondary lysozymes formed by the fusion of primary lysozymes containing engulfed molecules or organelles. Structure of lysozymes. The shape of lysozyme is irregular or pleomorphic, however, mostly they are found in spherical or granule structure. Lysozymes are surrounded by a lysosomal membrane that contains the enzymes within the lysosome and protects the cytosol with the rest of the cell from the harmful action of the enzymes. Functions of lysozymes. These organelles are responsible for intercellular digestion, where the larger macromolecules are degraded into smaller molecules with the help of enzyme present in them. Next one is mitochondria. Mitochondria are double membrane bound cell organelles responsible for the supply and storage of energy for the cell. The oxidation of various substrates in the cell to release energy in the form of ATP is the primary purpose of mitochondria. Structure of mitochondria. A mitochondrion contains two membranes with the outer layer being smooth while the inner layer is marked with folding and finger-like structure called cristae. The inner mitochondrial membrane contains various enzymes, coenzymes, and components of multi-cycle along with pores for the transport of substrates, ATP, and phosphate molecules. Functions of mitochondria. The primary function of mitochondria is the synthesis of energy in the form of ATP required for the proper functioning of all the cell organelles. Mitochondria also help in balancing the amount of Ca plus ion within the cell and assist the process of apoptosis. The next one is nucleus. The nucleus is a double membrane bound structure responsible for controlling all cellular activities as well as a center of genetic materials, and its transferring its one of the large cell organelles occupying 10% of the total space in the cell. Structure of nucleus. Structurally, the nucleus consists of a nuclear envelope, chromatin, and nucleolus. The nuclear envelope is similar to the cell membrane in structure and composition. It has pores that allow the movement of proteins and RNA in and outside the nucleus. It enables the interaction with other cells organelles while keeping nucleoplasm and chromatin within the envelope. Functions of nucleus. The nucleus is responsible for storage as well as the transfer of genetic materials in the form of DNA or RNA. It aids in the process of transcription by the synthesis of mRNA molecules. Next one is ribosome. Ribosomes are ribonucleoproteins containing equal parts RNA and proteins along with an array of other essential components required for protein synthesis. Structure of ribosomes. The ribonucleoprotein consists of two subunits. In the case of prokaryotic cell, the ribosomes are of the 70s with the larger subunit of 50s and the smaller one of 30s. Ribosomes are short-lived as after the protein synthesis, the subunits split up and can be either refused or remain broke up. Functions of ribosomes. Ribosomes are the site of biological protein synthesis in all living organisms. They arrange the amino acid in the order indicated by the tRNA and assist in protein synthesis. Next one is vacuole. Vacuole are membrane-bound structure varying in size in cells of different organisms. Structure of vacuoles. The vacuole is surrounded by a membrane called tonoplast, which enclosed fluid containing inorganic materials like water and organic materials like nutrients and even enzymes. These are formed by the fusion of various vessels, so vacuoles are very similar to vessels in structure. Functions of vacuoles. The vacuoles act as a storage for nutrients as well as waste material to protect the cell from toxicity. They have an essential function of homeostasis as it allows the balance of pH of the cell by influx and outflow of H plus ions to the cytoplasm. Next one is vesicles. Vesicles are structure present inside the cell that are either formed naturally during process like exocytosis, endocytosis or transport of the materials throughout the cell, or they might form artificially, which are called liposomes. Structure of vesicles. A vesicle is a structure containing liquid or cytosol which is enclosed by a lipid bilayer. The outer layer enclosing the liquid is called a lamellar phase, which is similar to the plasma membrane. One end of the lipid bilayer is hydrophobic, whereas the other end is hydrophilic. Functions of vesicles. Vesicles facilitate the storage and transport of materials in and outside of the cell. It even allows the exchange of molecules between two cells. Because vesicles are enclosed inside a lipid bilayer, vesicles also function in metabolism and enzyme storage. Next one is storage granules. Storage granules are membrane-bound organelles, also called zymogen granules, storing cells, energy reserve, and other metabolites. Structure of storage granules. These granules are surrounded by a lipid bilayer and are composed mostly of phosphorus and oxygen. The components inside these storage granules depend on their location in the body, with some even containing degradative enzymes yet to participate in digestive activities. Functions of storage granules. Many prokaryotics and eukaryotics store nutrients and reserve in the form of storage granules in the cytoplasm. Sulfur granules are characteristic of prokaryotic that utilize hydrogen sulfide as a source of energy. Thank you so much.